Welcome everyone to the fourth edition of the Tell Me More lecture series hosted by the Undiagnosed Diseases Network Participant Engagement and Empowerment Resource, or UDN PEER for short. The UDN, for those of you who may not be familiar, is a research study funded by the National Institutes of Health that applies interdisciplinary team science and cutting-edge genomics to provide answers for patients and families affected by complex medical challenges with no clear cause. The UDN PEER group is made up of patients and family members who participated in the UDN. And we work with UDN researchers and staff to improve the participant experience, to provide social support for families, and to share the UDN with others. My name is Megan Howley, and I have the great privilege of serving as the co-chair of UDN PEER. I'm also the parent of an adorable 7-year-old boy, Philip, who is a participant in the UDN and remains undiagnosed. Before I introduce our speaker today, just a quick housekeeping item. We have planned time after Dr. Green's lecture for questions, so please use the Q&A function and not the chat to enter your questions. And you can also vote for others' questions, which moves them up in the list. So if you feel so inclined, you can do that. And with that, it is my pleasure today to introduce our speaker, Dr. Eric Green. Dr. Green is the director of the National Human Genome Research Institute, or NHGRI for short, at the National Institutes of Health. He is the third NHGRI director, having been appointed by former NIH director, Dr. Francis Collins, in 2009. Dr. Green has been at the Institute for more than 25 years, during which time he has had multiple key leadership roles, including as the Institute's scientific director, chief of the NHGRI Genome Technology Branch, and the founding director of the NIH Intramural Sequencing Center. For over 2 decades, Dr. Green directed an independent research program that included integral, start-to-finish roles in the Human Genome Project and groundbreaking work on mapping, sequencing, and characterizing mammalian genomes. Dr. Green earned his MD and PhD degrees in 1987 from Washington University in St. Louis, coincidentally the same year the word genomics was coined. During his career, Dr. Green has authored and co-authored over 375 scientific publications, quite a legacy, to say the least. Dr. Green, welcome. We're so excited to have you, and the floor is yours.
Well, thank you very much. Let's get these logistics out of the way. And, oh, let me go back. You see my first slide, correct?
Yes, looks great.
Okay, perfect. Just wanted to make sure that we got that done. Thank you very much for that kind introduction. I am delighted to be here and happy to share with you my enthusiasm for and a lot of background information about this field of genomics, which I've been involved with essentially since the beginning of the field. Now, I fully appreciate the fact that I'm speaking to a likely pretty heterogeneous group. I think you all came to this group and you came to this seminar from different journeys. And I realize maybe your background about genomics or some of the concepts of genomics might be quite different. And I would imagine as part of the series, you're going to be exposed to lots of scientific accomplishments, lots of clinical stories, and all of it will be somehow connected to this field of genomics. So I thought what I would do is to really tell you by way of a foundational background, the story of genomics, in essence. And it's a story that can be told in about 45 minutes. And what this will provide is a fairly high level past, present, and future overview of the field. I'm going to emphasize the big questions and the strategies that have been used to answer them. And I think you'll see that this field of genomics, which now you find yourselves interacting with, really has had a metamorphosis over time. And despite its relative youth, the field has really unfolded as a very rich and exciting story. And that story can be told in a way that almost resembles a book in the form of chapters. And what's really important is, I think, as you'll see, is we now find ourselves at a critical juncture, as the title implies. We're just beginning to create the 4th chapter of human genomics. And I will also point out that the story that I'm going to tell you about human genomics is told from my perspective, and my perspective is one of a major funder of genomics research in the United States. As you heard, I'm now the director of one of the 27 institutes and centers that make up the US National Institutes of Health, Specifically, I direct the National Human Genome Research Institute. But both NHGRI and the National Institutes of Health have a major emphasis on health and human disease, and you'll see that will be the emphasis I will bring to the story as it relates to genomics. Now, to set up my book metaphor, I want to describe to you the table of contents associated with the story of genomics. And you will come to see that different parts of the table of contents nicely align with this trident-like graphic that you are shown here, which symbolically captures many aspects of the field of human genomics as it has developed over time. And the story of human genomics began with a prologue, which mostly reflected by something called the molecular biology revolution. Now, that prologue led to the dawn of the field of genomics in chapter 1, the era of the Human Genome Project, reflected a time when the field had a relatively focused scope as reflected by the narrow pink area of the graphic. Chapter 2 encompassed a time period immediately after completion of the Human Genome Project, when there was a rapid expansion and maturation of the field of genomics, as reflected by these orange arrows emanating outward from the central graphic. And following this period, there was a time of major transitions, as chapter 3 brought a greater focus on health applications, and the field was then en route to genomic medicine implementation. The prologue, in conjunction with chapters 1 through 3, actually represent the past. But as you're going to hear, we are currently creating the 4th chapter of genomics, which we aim to emphasize making genomics mainstream in medicine. And so during the rest of my talk, I'm going to provide for you how it is and that there has been this nice storyline of the prologue, chapters 1, chapters 2, chapters 3, and then into the future, chapter 4. And I'll provide details for each of these, although again, I'll be giving this all at a fairly high level throughout. And that really starts with the prologue. Now, for those of you who aren't very familiar with the origins of the science of DNA, it really grew substantially throughout the last century. Particularly notable events included the discovery of DNA's double helical structure, that iconic double helix, in the 1950s. And then later in the 1960s, we elucidated something called the genetic code, where we figured out how it was that DNA contained the information necessary for making proteins, the brick and mortars of our cells and tissues. But in many ways, the most relevant part of the prologue associated with the story of human genomics was the molecular biology revolution of the 1970s and the 1980s. And the big question for the prologue was, can we develop the tools for comprehensively exploring genomes? Now, the molecular biology revolution was specifically associated with the development of such tools. For example, those that could be used to isolate and to clone and to manipulate DNA. That is, these classic recombinant DNA technologies, such as those represented on the left. And later, methods for actually reading out the DNA letters, or in other words, sequencing DNA, such as the method depicted on the right. Now, don't worry about any of these details. The important things to know is that as the tools of molecular biology got better and better in the 1970s and the early 1980s, the notion of performing studies that would allow us to explore all the DNA of an organism, that is its genome, began to get serious consideration. And there was, of course, particular interest in comprehensively studying the human genome due to the compelling challenges in human and medical genetics that needed a better understanding of the structure and the function of the human genome, as well as more powerful approaches for actually studying human DNA. In short, the advances in the tools of molecular biology and the growing excitement about the new approaches for comprehensively studying the DNA of an organism led to the coining of the word genomics, and also the naming of the new field of genomics, both that occurred in 1987, as you heard in my introduction, was the year I graduated from medical school and graduate school. And in fact, I can tell you that you will not find the word genomics in the medical or scientific literature prior to this lead editorial in a brand new journal that got created in 1987 called Genomics, in which the editors wrote that for the newly developing discipline of genome mapping and sequencing, including the analysis of the information, we have adopted the term genomics. And so genomics has now been a word in our vocabulary for the past 35 years. But it was really not an accident that the word genomics was coined and the field of genomics was established in the late 1980s, because both of those two things were related to the early planning for a major effort to map and to sequence the human genome for the very first time. And of course, that effort would eventually become known as the Human Genome Project, reflected by Chapter 1 in the Story of Human Genomics, which covered the years 1990 to 2003. Now, the big question for Chapter 1 and the Human Genome Project was, can we map and sequence the human genome? Now, I should pause here and make an important point. Each of the 4 chapters of human genomics that I'm going to describe was not written by a single author. It did not play out under the guidance of a single country or a single funder, but rather it involved strategic visions that were developed by multiple members of the genomics research community in conjunction with research funders. And these guides provided a blueprint, if you will, for the pursuits of human genomics during that time. And for example, I can tell you the Human Genome Project was guided by 3 successive strategic visions shown on the left, which were instrumental for articulating and when needed, re-articulating the goals and the plans for the Human Genome Project. Well, by all accounts, the Human Genome Project was wildly successful. It accomplished all of its goals ahead of schedule, and it provided the world the first reference sequence of the roughly 3 billion letters that make up the human genome. I got involved in the Human Genome Project shortly after I had gotten out of medical school and graduate school. I got to be involved in a research laboratory that was developing some of the earliest tools for mapping and sequencing human DNA. And so I got involved literally on day 1 of the Genome Project, participating in it and was involved throughout. Now, there are a number of things to tell you about the Human Genome Project, which you could read about in the history books. But at the time, it was very unusual to have a big science, consortium-based, highly managed project in biomedicine. That's just not the way biology was done back then. We didn't do production science. It also was very unusual to have one big project that was internationally organized and involved multiple funders. It was also unusual to have highly focused goals in a biology project. And it was also very unusual for a biology project to be very technology oriented. All of those things made the Human Genome Project at the time controversial. However, it was compelling, it was hard, but it was successful. And so even though it wasn't guaranteed that it was going to reach its goals, and at times I can tell you it was quite scary, at the end of the day, the Human Genome Project succeeded in all of its goals and wrapped up in 2003. And by 2001, I can tell you a draft sequence, the first draft sequence of the human genome was published in these 2 journals, Nature and Science. There was a public effort of the Human Genome Project in Nature, and then a company had started to try to sequence a human genome, and they reported their work in this issue of Science. And in fact, these iconic journal covers took place in 2001, and both of those journals commemorated the 20-year anniversary of this by publishing lots of things about genomics last year to commemorate the 20th anniversary. Oh, and by the way, the Genome Project ended in 2003. So spoiler alert, next year is going to be 2023, and I can guarantee you we will be commemorating and celebrating the 20th anniversary of the completion of the Human Genome Project. So stay tuned next year for a lot of 20th anniversary commemorations and celebrations. Well, what were the main highlights of Chapter 1? If I was going to give you in sort of the Cliff Note form, what are the things to remember of Chapter 1? Well, the main highlight was we generated the very, very first sequence of the human genome. We generated it. We had at least a representation of a human genome, roughly 3 billion letters ordered out. There were other highlights. We learned how to share data better. There was a lot of data sharing that was implemented for the first time as part of the Human Genome Project, which continues to today. We integrated a lot of bioethics into biological project. You may hear about ethical, legal, and social implications research, or ELSI research. This became prominent during the Human Genome Project. And I think grew out of this a heightened respect for consortium-based science and biology, something that biology didn't do much before that. So those were the highlights of Chapter 1. But the completion of the Human Genome Project marked the end of one chapter in human genomics, but the immediate beginning of another. And Chapter 2 saw the field grow well beyond the focus goals of the Human Genome Project, bringing with it a major expansion and maturation of genomics. And in fact, upon completion of the Human Genome Project, I can tell you many applications of genomics immediately arrived on the scene. In fact, shown here are 9 representative areas in which genomics quickly came to play a major role. And each of these represents a new and interesting story in their own right. But the storyline for genomics that I am going to tell you about, of course, is focused more on applications of genomics to health and to disease and to medicine, especially because of my institute's and the National Institutes of Health's emphasis on human health. Well, the big question for chapter 2 was, can we use genomics to advance knowledge about health and disease? And with completion of the Human Genome Project, the various funders of genomics research actually went in various different directions to pursue their different missions. In fact, some of them went to support research in the 9 areas I just showed you on the previous slide. NHGRI recognized the importance of having a well-crafted vision for supporting human genomics research beyond the Human Genome Project focused on human health. And as such, our institute conducted a multi-year strategic planning process and timed things such that literally the day the Human Genome Project ended, NHGRI published this strategic vision that charted a course for the immediate future of genomics beyond the Human Genome Project. And the iconic illustration from the strategic vision is shown on the right that depicted how human genomics would now be built up upon the foundation created by the Human Genome Project. Well, the 2003 NHGRI strategic vision then guided efforts in human genomics until 2011. Well, improving human health was always a key rationale for the Human Genome Project, and each of us has a unique genome, and knowledge of the specific features of each patient's genome might provide the means to actually improve the practice of medicine. And so guess what? It was sort of obvious that when the Genome Project ended, the idea of somehow marrying genomics and medicine together was rather obvious. And in fact, the popular press, such as New York Times Magazine, or the scientific press, such as Science Magazine, they started to use this phraseology of genomic medicine. It was the obvious thing to do. The problem is that while we could say those two words together, it was a rather blurry what that actually meant. We could envision somehow how the use of genomic information as part of the practice of medicine, but that was far from being in focus. But what was envisioned was being able to encourage and develop a medical discipline that would involve using genomic information as part of clinical care, and then considering how that might be influencing the practice of medicine. And of course, the reason for this is that all of us has a slightly different genome. Each of our blueprints is slightly different, and yet clinicians are blind to that. But we know that some of those differences influence health and disease. If we could somehow capture information about our genomes and use that as part of medical care, that would be genomic medicine, and perhaps we can get better at the practice of medicine. I can tell you that virtually everything I was taught in medical school was based on what worked for the average patient. The problem is that none of us is average. Each of us is unique, and could we capture the uniqueness as a means to tailor our medical care? You will hear other phrases like genomic medicine. You will hear phrases like personalized medicine or individualized medicine or even precision medicine. Genomic medicine is just a much more narrow construct. It's just talking about uniqueness in our genomes, whereas personalized medicine, individualized medicine, precision medicine takes into account personalization of lots of things in our lives, even beyond our genomes. So genomic medicine just focuses on genomic differences among us that might be useful in the practice of medicine. Well, how are we gonna get to genomic medicine? What does that really mean? I mean, the Human Genome Project was a journey. It was really the first journey in the story of human genomics. But realizing genomic medicine really should be viewed as the second journey in the story of human genomics. And we really had to recognize that the Human Genome Project was one finish line in one way, but really was the starting line for the next journey. And that second journey, which would start upon completion of the Human Genome Project, was ultimately going to attempt to realize genomic medicine in some meaningful way. And just like the Human Genome Project, this was going to involve multiple steps. and multiple progressive accomplishments. We wouldn't even know what all of them were. But once we started down this journey, like the Genome Project, as we accomplished things, we could check them off the list. We could say, yep, got that done. Yep, got that done. Yep, got that done. And so as we thought about this journey towards genomic medicine, we also recognized that, like the Human Genome Project, this was not going to be done by one researcher or one clinician. It wasn't going to be done by one country. It wasn't going to be done even by one group of individuals, nor was it going to be a sprint. Rather, it was going to be much more analogous to a marathon. Many people, many diverse, different people, different expertise running shoulder to shoulder for a long time and eventually attaining some goal. But it was going to be a lot of work. Well, what were some of these steps? Well, I can tell you that the very significant barrier that existed at the end of the Human Genome Project was the cost of sequencing a human genome. And we knew that the very first thing we needed to work on was to reduce that cost so we could go from just having one human genome sequence to actually eventually being able to have genome sequences of individual patients. Well, let me put a fine point on it. The Human Genome Project, wildly successful, generated the first sequence of the human genome. But that first sequence cost about $1 billion. It was a great investment of $1 billion, but $1 billion was for the Genome Project. It was never going to be for genomic medicine. To make genomic medicine a reality, we would need to reduce the cost substantially. We would need to knock a lot of zeros off of that figure. In fact, we always projected we would need to get technologies that will allow us to sequence a human genome for something like $1,000, the cost of maybe an MRI or a CAT scan, a very reasonable cost for a major clinical test. In fact, we proposed this as being needed the day the Genome Project ended. We said we needed a $1,000 genome. You may even have heard that phrase, the $1,000 genome. It was really basically a battle cry to come up with new ways of sequencing DNA that could deliver a human genome sequence for $1,000. And we knew that the ways we sequenced the human genome the first time by the Genome Project in facilities like that shown on the slide were going to have to be retired. It was going to require something far less expensive than these kinds of factories that cost millions and millions and millions of dollars to build up and operate. Rather, we were going to need some science fiction-like device that could very inexpensively sequence a human genome. Well, let me just fast forward and tell you the rest is history. We've done it. In the 19 years now since the end of the Genome Project, we have seen a precipitous decline in the cost of genome sequencing because of all new technologies that have basically supplanted the technologies that were used in the Human Genome Project. I don't have time to talk about this in detail, but it now costs less than $1,000 to sequence a human genome. So check, we've gotten that first step out of the way. With that, it meant that now we had the ability to start to study how each of us is unique. And I talked about how our genomes vary from one another. And we have now gone from having one genome sequence to having literally millions of genome sequences. We can compare sequences to one another, and we could find those places in our genomes that we are different. And that has been profoundly valuable from having a better understanding about human genomic variation among people, across the world, in different countries, different populations, and so forth. And so that's another and very important step. You could sort of say, check, we've gotten a lot of progress in understanding how our genomes differ. In fact, most of us are 99.9% identical at the sequence level. And then we have a lot of information about what those variants are, where they're located, and so forth. We've also learned a lot about how the human genome functions. The Genome Project was about just giving us the letters of DNA. It was not about giving us an interpretation of those letters. And here I could talk for hours about the complexities of the human genome sequence. And I don't have time to talk about the 19 years of progress we've made in interpreting those complexities. But needless to say, as represented on this shot of this graphic that represents how you could go on the internet and zoom in on browsers that give you lots and lots of GPS-like information about where are the functional sequences in the human genome, whether they be genes or whether they be sequences that function in other ways. Needless to say, while we have a long way to go in fully interpreting the 3 billion letters of the human genome, we have made substantial progress in understanding it. And that has given us an incredibly valued ability to be able to go and look at somebody's genome and begin to figure out where do they have variants, and how might those variants influence people's genome with respect to how it functions, and whether or not those variants are relevant to people's health and disease. So when I summarize chapter 2, I would tell you the highlights really relate to how we have expanded and matured the field of genomics. The most important highlight were these new technologies that deliver the $1,000 genome. But other important technologies, we developed an emerging catalog of literally millions, tens of millions of known places in the human genome that people vary. We know what those variants are, and increasingly, we are learning what those variants mean. And we're doing that because we have progressively identified functional elements in the human genome and figured out much about the incredibly complicated choreography that takes place among those 3 billion letters. And much of this has been driven by improvements in computer science and data science and bioinformatics. Those are now central to genomics and really became so throughout chapter 2 into chapter 3 and beyond. So that was chapter 2, and that takes us back to about 12 years ago, because by around 2010, progress in human genomics was really quite impressive, so much so that it became time to more aggressively pursue applications of genomics to medicine. And so chapter 3 was very much about beginning to explore the clinical applications of genomic tools and resources, putting the field en route to genomic medicine implementation. And the big question for chapter 3 was, can we demonstrate effective uses of genomics in medicine? And so once again, to prepare for this chapter, our institute once again did a multi-year strategic planning process where we engaged lots of people in the community to help us chart a course towards genomic medicine as we progressed essentially from base pairs to the genome, as exemplified by this strategic plan that we published in 2011. And it really was this progression from base pairs to the bedside of patients that was iconically represented in the central graphic of this strategic vision shown on the right, which really just represented how we were going to move along from basic knowledge of the genome all the way to the care of patients using genomic information. And so this strategic vision then guided efforts in human genomics for the rest of the decade. Now, key to the next step in this journey was really understanding the relationship between genomic variation and genomic function. And as I sort of intimated, not all genomic variants are biologically consequential. Only some genomic variants really have any biological meaning. And some of those actually influence our traits, or in other words, known as our phenotypes. And the traits that I know most of us care the most about are those related to human disease. And what really became very clear is that we were poised in chapter 3 to really accelerate our understanding of the genomic basis of human disease. And to really appreciate where we have made those strides, it's important to keep in mind the general framework that we use. It's an imperfect framework, but it generally works with respect to understanding the architecture of genomic diseases. I think what this organization is all about, and what probably most of you are the most interested in, are rare diseases. These are diseases that are rare in the population, but which are generally caused by mutations or DNA changes in a single gene. And that is the major reason why an individual might get that disease. These are also known as monogenic or Mendelian disorders. And here, chapter 3 brought remarkable progress. And in fact, today, we now know the genomic basis for nearly 6,000 rare diseases. And you only have to go back to the beginning of the Human Genome Project, 1990, when that number was 61. So from 1990 to now, we've gone from 61 to nearly 6,000. Now, there still are some rare diseases, several thousand, that we don't yet know a genomic cause. But I would say among the incredible highlights in genomics, our advancement in understanding the genomic basis of rare disease really deserves remarkable credit in terms of an accomplishment. Now, most diseases that fill hospitals and clinics around the world are not rare, but rather are common. But the diseases shown on the right are represented as common diseases because their complexity resides in their multifactorial nature. There is often a greater number of genomic variants that contribute to the disease And there's often a greater contribution of environmental, lifestyle, and other factors to the disease. So it's much more of a gene environment interaction situation compared to rare diseases. And here, there's been major progress, especially in statistical associations that have given us lots of clues of where to look in the genome. But really, the great advances we believe in common diseases are still in front of us as opposed to in the rearview mirror. And so I think we can marvel at our progress in rare diseases, and I think we're poised to make major progress for common diseases. Well, by the middle of the last decade, things had really started to settle in with respect to efforts to apply genomics in medicine. I mean, we had cheap genome sequencing, we had catalogs of genomic variants, we had functional understanding of the human genome, and we had made major progress in disease gene identifications. And these developments culminated in some of the earliest examples of genomic medicine being implemented. And while it's just the tip of the iceberg with respect to how genomics might eventually be used in medicine, these examples brought clarity to the situation, bringing genomic medicine into focus for the very first time. Now, what were some of these early examples of genomic medicine that were being implemented? Even if they were just the earliest examples, I think these are worth pointing out because they really represented highly illustrative circumstances where the practice of medicine was being changed because of genomics. Each of these could be a talk in and of themselves. Among them is, of course, cancer genomics. Cancer is a disease of the genome, and genomics has just completely changed the face of cancer research and increasingly cancer diagnostics and hopefully eventually cancer treatment. What I know this audience is most interested in, of course, is rare genetic disease diagnostics in particular. And here, it was just remarkable what Chapter 3 really brought, the last decade has brought. And of course, I don't need to tell this group about the Undiagnosed Diseases program that started here at NIH that eventually morphed into the Undiagnosed Diseases Network, which is the basis for the UDN PEER group. And NIH has been a big enthusiast. I have spent countless hours helping do things to facilitate advances in the Undiagnosed Diseases program, more recently, the Undiagnosed Diseases Network. And it really represents a triumph of personal accomplishments by people who were advocates and physician scientists poised to bring genomics to rare diseases, and also an incredible partnership with families and patients and new insights of ways to use genomics to be brought to bear on very challenging problems. And of course, it also involved team science, consortium science, data sharing, a lot of the fundamental things that are so important in genomics to bring together the success of the UDN. I hope you also appreciate that even my institute, things we've been involved in, we've been a major leader on behalf of NIH for the UDN. But undergirding all of this were also major programs in rare disease genomics, such as our Centers for Mendelian Genomics, which more recently has morphed into the Gregor Consortium, which is all about accelerating progress in understanding the genomic basis of rare diseases, and also very much involved in efforts to operationalize the use of genome sequencing as part of clinical care. And we even did this previously in partnership with the Child Health Institute as part of a newborn sequencing research program that yielded most substantially the use of rapid genome sequencing in the neonatal period, which is now being operational. It's one of the great examples of genomic medicine being implemented, being operationalized in neonatal intensive care units, really around the world, but certainly across the country. And so this has been a major success story of genomic medicine, the use of genomics for rare genetic disease diagnostics. In addition, there's been success in the use of genomics for better prenatal genomic testing, especially non-invasive prenatal genomic testing. Also significant advances in genomics being used for the selection of medication, something known as pharmacogenomics, where we better match patients to different types of medications, the ones that they may be most effective for those individuals. And a lot of attention is being paid to the use of genomics for more common diseases to try to predict risk, not by knowing the exact genomic basis with respect to genes, but just being able to calculate based on the signatures of genomic variants someone might have, whether or not that puts them at greater risk for common disease or not, I'll put a question mark around this because I'm not sure we've really implemented this yet. It's still part of a research study, but I believe it might be the next one to be implemented in reality with respect to genomic medicine successes. So when I think about chapter 3, what were some of the main highlights that really saw the field move towards having genomic medicine become a reality? Clearly, the compelling examples I just laid out for you, where genomic medicine was no longer hypothetical, it was real. Some of this represented major progress in unraveling the genomic basis of rare disease, some of it in providing a framework for more common diseases. And I think some of it has just seen a very widespread dissemination of genomics across all of biomedical research. Well, thus far, I've told you about the past and to some extent the present. I should pause and take stock and really remind you what we should be celebrating. I think it is worthy of celebration when we consider the progress we've made in the past 3+ decades. Think about it. We generated the first human genome sequence by the Human Genome Project. Check. We went on and reduced the cost of sequencing a human genome and reduced the cost by more than 1 million-fold. Check. We were able to then sequence millions of human genomes, not just have one human genome sequence. Check. We then went on and had profoundly advanced our understanding about how the human genome functions, and then proceeded to use that information to significantly advance and unravel the genomic basis of human diseases, and particularly rare diseases. And finally, we actually came up with real examples of genomic medicine being implemented. And for all of these 6 major highlights, I think it is worth a celebratory note. At the same time, and before I pivot to chapter 4, I will point out we're not perfect. We still have challenges, and we really have some major things we are wrestling with now and will continue to wrestle with in the future. We still can't immediately go from having a genome sequence to know clinically what it all means. There is still a hurdle, and some of you may be facing such a hurdle where you have a genome sequence that you're interested in trying to figure out everything about, and you represent how complicated all those differences are. So that still represents a major challenge. We have many societal challenges because genomics is now relevant to society. Genomics is touching healthcare. Healthcare is one of the most complicated societal ecosystems that we deal with. We're facing complexities of coverage and payment, complexities of privacy, complexities of regulation. And so all of the things that we always face when we deal with healthcare, now genomics finds itself in the middle of. So those are things we are now grappling with. We have some problems with diversity. Too much of our successes in genomics have been a result of the study of individuals of European descent. We have an embarrassment of not sufficiently paying attention to diversity over the years. We are trying to fix this, and it is a high priority, as you will see coming up in chapter 4. And finally, we also have to recognize that genomics isn't the answer to everything, that there are both genomic and non-genomic factors in human health and disease. And while we may have great successes in genomics, we need to also bring along successes in identifying the non-genomic factors and how some of these may interact with the genomic factors if we're really going to understand comprehensively human health and disease. Well, that's the past and the present so far. So as we approach the year 2020 and the start of a new decade, it was apparent that progress in human genomics continued to be breathtaking, as exemplified by the 6 celebratory areas of progress that I just highlighted. And in particular, that we were beginning to see the earliest successful applications of genomics. But we also realized that our strategic vision that guided us through chapter 3 was about 10 years old, which is a long time in genomics. And we needed a new updated vision. And if we were going to really take advantage of creating chapter 4 in this coming decade, we would need to develop a new strategic vision, one that would help us make genomics mainstream in medicine. And so our main question about the future, and the big question for chapter 4 was, can we deploy genomics broadly and equitably in medicine? And so we did another round of that strategic planning that we've done previously, and here's our latest strategic vision. And so I now want to spend the rest of my talk describing this 2020 strategic vision, which I hope will provide you a preview of how Chapter 4 is being created and what human genomics might look like in the coming decade. Well, one of the things to really appreciate about the current decade that's different than the previous decade is that genomics is now everywhere. It's not just a little field over here, it's woven throughout the fabric of everything going on in biomedicine. You know, once upon a time, genomics was just about the Human Genome Project, which amplified our view of that double helical structure. But once the Genome Project ended, everything I've told you about in the last 19 years were the threads of genomics getting increasingly woven into basic science research, into translational research, increasingly into clinical research, and nowadays a diverse set of people continuing to weave genomics into medicine, into society, and into really everything that's going on in biology. And that meant that we could no longer strategically plan for all of genomics, but rather we were really going to have to really focus our attention. And it was actually ironic that The Economist wrote a celebratory article about the Human Genome Project. Literally at the time I was working with the graphics artist on the graphic I just showed on the previous slide. And in this article, they specifically said genomics is now so embedded in biology that it's hard to recall what things looked like before it. That's exactly as we felt when we started this new round of strategic planning. And so to account for that, we realized that we were going to have to redefine our identity as an institute, and we were going to redefine what we were going to do. We could no longer provide a strategic vision for all of genomics, but rather our organizational mantra and our mindset, and for the strategic vision, was to focus on the forefront of genomics. And in doing so, we would talk about the cutting edge, the front end, however, the tip of the spear, whatever metaphor you want to use, and that would guide our strategic planning effort, which ultimately led to this strategic vision. And if you look at this URL on the right, if you have not read these 10 pages, please go to this website or email me. I'll send you a PDF. Please read this. It is written for a very general audience. And I can't do it justice in the next 6 or 7 minutes, but the 10 pages really very nicely summarizes all the exciting things going on in genomics that you could look for in the coming decade. Now, what was interesting also, and I'm going to just go through this very quickly, is that what we heard when we did strategic planning this time was that what was needed at the forefront of genomics was not all big research projects. That's what we were used to. Rather, it was very heterogeneous what we heard about. We heard about multiple different kinds of things that we needed to provide at the forefront of genomics. And at the end of the day, it could be summarized as responsible stewardship. That for us as a funder, to be at the forefront of genomics meant taking responsible stewardship of a number of areas. What were those areas? First area were guiding principles and values for human genomics. Being at the forefront of genomics meant that we needed to be responsible stewards of the guiding principles and values that should be used in doing human genomics, the current ones that need to be re-articulated and future ones that need to be designed. Some of these related to diversity, some of these related to equity and social justice, some of these related to consortium-based team science and open science and data sharing, some of these related to things like data standards. But these were all important things. They weren't research projects, they were principles and they were values. A second area of things that we heard that represented responsible stewardship at the forefront of genomics was we needed to take responsibility for the foundation of genomics that everybody uses. We needed to sustain that foundation. We needed to improve that foundation. We needed to make sure that foundation was robust. And so there's a series of elements of this foundation that we are now going to be responsible stewards of. Some of these relate to genomic data science, how we generate data, how we manage data, how we have data resources. Some of it relates to comparative genomics, comparing our genomes to other animals and learning from that. Also, some of this relates to a foundation that provides individuals an empowerment to be stewards of their own genomic information. And some of this reflect things such as genomic literacy and the training of professionals in genomics, not just individuals in the public and patients, but also healthcare providers. A third element or area that was important at the forefront of genomics were recognizing that there are barriers that impede progress in genomics. And this was deja vu all over again, because 19 years ago, when the Human Genome Project ended, the big barrier in front of us was the cost of DNA sequencing. That's why we had the $1,000 genome effort that yielded a millionfold reduction of the cost of sequencing DNA. Responsible stewardship at the forefront of genomics is about identifying the next set of barriers, knocking them down, and everybody would benefit from that. The kind of barriers that could be envisioned, some of them relate to synthesizing and editing DNA. Some of them relate to characterizing genomic variants or dealing with complex biological processes like genomic mosaicism, or dealing with things about implementation in healthcare. Healthcare is a barrier in and of itself, and there's a science of how best to implement things in healthcare. So some of this had to be breaking down these barriers. And finally, some of the things we heard related to developing compelling research projects in biomedicine, and responsible stewardship in genomics. At the forefront of genomics represents carrying out those research projects. Some of those will relate to comprehensive views of genes and regulatory elements, continuing to study the genomic architecture of human diseases and traits, both rare and common, enhancing diversity in genomics research, thinking about things beyond genomics, other omic types in clinical settings, and also thinking about how you could use healthcare systems as learning environments for implementing genomic medicine. So that is the new strategic vision. I didn't do it justice. Please read it, the 10 pages, because you will hear in greater detail the elements that make up each of these 4 areas that together provide an organizing framework for seeing the forefront of genomics bring us the kind of advances in the coming decade that we are expecting. And finally, we also went out on a ledge a bit, because we ended our strategic vision document by making 10 bold predictions about what might happen in human genomics in the coming decade. Now, these predictions were intended to be inspirational and aspirational. We really looked at them as high risk, high reward kind of predictions. We don't think they're all going to come true, but they really serve as something to reach for. And so I thought I would sort of, just near the end of my talk, share with you a video we put together, which very much summarizes these 10 bold predictions, and I think gives you a little bit of the spirit of what we were trying to do in putting together a group of bold predictions as we think about what chapter 4 might look like. So watch and listen to this video. This is just a short video.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6, genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7, it will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8, a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9, advances from human genomics will benefit all of society and not just a few. Prediction 10, genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So if you liked those 10 bold predictions, we actually just wrapped up a seminar series we had where we took each of the predictions and we had speakers come in and talk about them, and we videotaped all these. And so shown here is a URL, and I'll show you another way you can get to it later. If you're interested, you could watch any of these 10 seminars to learn more about these bold predictions. So what we basically did is we put together a strategic planning process that fed in ideas, needs, proposals, and obstacles, and we synthesized it into the 10 pages that we published in Nature of our 2020 strategic vision that I hope you will read. But of course, now attention is shifting as we are creating the 4th chapter by extracting a subset of the ideas in that strategic vision. And from that, we will fund projects and organize programs and launch new initiatives and develop new policies. And that's what we're busy doing. And there's lots of things happening I don't have time to talk about. I will point out that one of the areas we did jump in almost immediately after the strategic vision came out in early 2021 was an emphasis on trying to improve the diversity of our workforce. And this all relates to a set of issues we have in genomics where we are simply not diverse enough. We made as one of the highest priorities a long-term proposal to enhance the diversity of the genomics workforce. And we think this is very important for a number of reasons, which I'm happy to talk about in the discussion. And so I just point you to this as an example of something being launched directly out of the strategic vision, but many other things are happening as well. So in closing, I really hope you've enjoyed hearing at least my version of the story of human genomics, one that has certainly been seen through the lens of a funder of this remarkably exciting field. You know, the prologue and those first 3 chapters have now been written, but hopefully the most impactful parts of the story in terms of improvements in human health are still to come, especially work at the forefront of genomics that aims to use genomics to really see it become mainstream in medicine. And I certainly can assure you that the institute that I lead and the community that I represent is just working tirelessly to see that vision realized. So lastly, if you enjoyed interacting and hearing from me, we really make it a big priority to help people stay connected with us. We have this new webpage we just set up. It's a very simple URL. If you go to that webpage, genome.gov/stay connected, you'll see this. If you want to not look at my face, you could scroll down and you'll see this. And here you can get very easily by clicking on any of these squares, access to our whole website, some newsletter I put on, or you want to follow me on Twitter. I have a very, very active Twitter account. Follow me or get a newsletter from me. You could read more about our strategic vision or our brochure, or I told you about some videos in various ways. We have a whole Genome TV channel at YouTube. If you were interested, and you could find that as well. And if you scroll down even further, you get to some select videos that my communications group picks if you want to see other talks of mine, or also get to some podcasts and op-eds that I've written. And with that, I will stop. And I know I went a few minutes longer than I thought I was going to go, but I also don't have a hard cutoff at the top of the hour. So I will happily stick around to answer your questions for those who want to stay overtime. And I will stop sharing my screen.
Great. Thank you so much, Dr. Green. That was quite a tour de force of the history of genomics. it's quite overwhelming, the progress that's been made over the last 3 decades, and you summarized it so nicely. We have a lot of questions, but I'm gonna try and combine a few of them because there's some themes, particularly around next steps for undiagnosed and rare disease, which is really, many in the audience are very interested in this. And a lot of the questions center around what's missing, what are we missing for those who are undiagnosed. You know, the Undiagnosed Diseases Network, as you talked about, has been incredibly successful in diagnosing 35% of these really tough cases. And there seems to be a lot on the horizon. We had questions about non-coding regions of the DNA. We had questions about epigenetic changes in DNA methylation, which I don't even know what that is, but that sounds like an interesting point as well. We had questions about long reads. So I'm wondering if you would speak a little bit more to how your vision for the future is, your specific interests and your vision for the future specifically in that area.
Yeah, no, these are great questions. And what I think the best way to give a broad answer that is it really represents why it is so important that we continue to, that we don't sit there and pick up our efforts and just move them to the hottest area, but rather we must maintain a commitment on all areas. And by that I mean, because some of the things you just mentioned, we need to continue to interpret and reinterpret and constantly characterize the human genome. And while that is because there's so much more to be learned. And so there needs to continually be an investment in very basic science so that eventually we will understand base by base every part of the human genome, and we're a long way from that. Similarly, we need to continue to invest and develop even better technologies for studying all sorts of things about the human genome, not just its sequence, but you start to touch on epigenomics or ways our DNA can be modified. And so in doing so, we might learn of other ways in which genetic diseases can manifest themselves. But in order to do that, we need better technologies. Some of those are going to be laboratory, some of those are going to be computational. So it really represents, a continual, not abandoning, or even thinking about, by the way, coming out late in about a month or sometime over the next month, there'll be a lot of excitement that we finally have figured out the sequence of every base of the human genome. You may have thought, oh, well, didn't the Genome Project do that? Well, actually, there were parts of the genome that were not even sequenceable at the time of the Human Genome Project. But we finally figured out how to sequence those. So when you're dealing with undiagnosed condition, you're not certain, but you don't know whether you're not reading the right letter, or you don't know whether you don't understand the function of that letter, or whether it's not a letter difference, it's another kind of a difference. And so that is exactly why we need to continue to push on all fronts. And some of these things will bear fruit and some of them won't, but we don't know until we go there, which is why you need a full continuum of investment from very basic science, computational, laboratory technology. You need some of it to be focused on the disease. You need some of it to be focused on medical applications or therapeutic development. You need a diverse portfolio.
Thank you for that. Thank you for that. I just received word from our Rachel, that we have someone from the UDN, Dr. Sesh Cole, who's one of our wonderful PIs, who would like to ask a question.
I'm going to embarrass Sesh by first of all, I'm intimidated by being asked a question at Sesh, because the last time Sesh asked me a question, I was a third-year medical student, and he was an attending in the neonatal intensive care unit, and and he's so much smarter than I am when it comes to medicine. So I'm completely intimidated by taking a question, but I won't embarrass him by telling him what year that was, although you guys could do the calculation.
But I'll only say back that I was 3 years old. That's how I can be so young.
If I gave you the year, you would think Sesh was 5 years old or something like that. What's up, Sesh?
Eric, really terrific talk and really terrific leadership at NHGRI. And I think all of us in the medical and genomic communities are grateful for your continued leadership and vision. I would put in a plug in terms of aspirations over the course of the next 10 years on behalf of children to be sure that we are acknowledging the importance of development for genomics, because a variant in the genome may have different impact in a 1-year-old or a 10-year-old or a 20-year-old. And making sure we're acknowledging and intentionally investigating that interaction between development and genomics will be very important, I think, for children.
And Sash, and even beyond that, right, you'd also point out, you're giving postnatal development, and the same complexities probably apply to prenatal development, right?
Correct. And we're very anxious to be sure that the work that's been done now on prenatal application of genomics to evaluation of pregnancies is continued and accelerated. In fact, prenatal genomics is one of the most rapidly expanding areas in genomics and the most rapidly applied areas in genomics. And we really need to intentionally, again, to focus on what the impact of those variants is on fetal and postnatal development, and quite frankly, how they help us understand the origins of adult diseases.
Yep. I couldn't agree more. I will tell you, I don't want to get too geeky with various new programs we're launching, but in partnership with the Child Health Institute here at NIH, it's a perfect example. And as taxpayers, you should all be happy to hear this, that you have two parts of NIH getting together and investing funds together in exactly what Sesh was just saying, to really try to understand genome function at different stages of development. It's a project called Developmental GTEx. I don't want to get too geeky, but it's exactly the mainstream of what of Sesh's point was, and I completely agree with it. And it really relates to the complexities of the genome. You know, we think of it as, you know, these 3 billion letters, and there's some no. There's so many different dimensions over time, different developmental stages, different tissues exposed to different things, et cetera, et cetera. It's very complicated, but it's also very exciting. We continue to hope that lots of people will be attracted to the field. It's not getting less boring. It's getting more and more exciting every year.
Thank you, Sesh. I know we're over the top of the hour. Do you have a couple more minutes?
No, no. Just as long as people want to stick around, I will keep going. I really.
Okay, we still have almost 100 people signed on. So
no. You know, the people should. If they've got things to go to, they should go to it. But I just assume not leave any question behind. I'm happy to stay here.
So this is a really interesting one that came in from someone who's asking about what the UDN and other programs moving forward can learn from big successes like the Human Genome Project, and particularly how the HGP was able to overcome kind of old school scientific goals versus new investigator innovations, and some of those tensions, competition among investigators. You know, what can we learn thinking about the future from those experiences in the past?
No, it's a great question, and it's actually something I've written about and others have written about. Is, you know, sometimes we only talk about the tangible products of the Human Genome Project, you know, the sequence, the technologies. I often emphasize there was a lot of cultural contributions. And I mean, I mentioned some of them in my talk. I did it very quickly, but, you know, again, the idea that, and UDN exemplifies it, I mean, beautifully, that, you know, team science is so much better for hard problems like what UDN faces. Team science is orders of magnitude better than individual people's science. Similarly, data sharing. You know, the second people start to get too protective of their data, everybody suffers. And the whole idea of open science and data sharing, incredibly important and incredibly powerful when you have it. So I do think there's a lot of stylistic things that we have learned from the Genome Project. It doesn't mean that all science is done that way, but there are a number of pursuits that science is best done that way. And you don't have to be afraid of it. And it could be incredibly beneficial to everyone involved. And so I think we have learned a lot, and you know, and in the flip side of it is, I think we've also learned, and I think one of the things UDN has helped, and other similar projects, you know, is that I think us scientists learn that, you know, we don't know everything. We need to talk to the patients and the patients' families and the communities, and that engagement is also very important for navigating a lot of complexities related to the interface between genomics and the actual use of it in medicine.
Great, thank you. Another question we have, again, relates to kind of looking ahead. You know, everyone who's familiar with the UDN is aware that it's reaching the end of one phase and looking forward into the future and what that next phase will look like. And we have a question from a participant who, I know this person personally, as someone in the UDN, is undiagnosed, who's saying, what's your advice for those who are undiagnosed who are in the program? What should their next steps be? You talked about some of the exciting technological advances, but kind of on a practical level, if you're a participant in research and you want to contribute to research, but you don't yet have answers, where would you suggest people go?
Well, I would sort of say if you're in the program, you know, the bottom line is stay connected with the people that you've been interacting with, and because you know the people that you've interacted with the UDN are desperately trying to be as helpful as they can be, and are very, very interested in doing whatever they can do within their power, to not only for you, but for others who are in similar circumstances. And, you know, I think increasingly it's very important as we continually have to navigate the complexities of bringing genomics into medicine to make sure that patients' voices are there at every level. It is a complicated ecosystem, and we have it's totally crazy in the U.S. and then you look at any other country and then it's crazy in a different way. It's like nobody has really solved healthcare payment, et cetera, et cetera. So I just think, but it can't just be scientists yelling at healthcare, payers or government officials. It has to be, we need the whole community trying to reason through how are we going to integrate these things, and the patient's voice are very important there.
Well, thank you for that, and I think it's certainly something that the UDN peer group, hopes to do,
Right.
to partner and support this work moving forward. We have another question about the transition from diagnostics to therapeutics, and it wasn't as much of a focus of your talk, though it was certainly woven within there. But someone's asking, it's really exciting to learn about the advances in diagnostics, but what do you see as kind of the next steps for transitioning that to therapeutics? And they also ask, and this relates to what you were just saying, how do we work together as scientists and advocates? There may be patients who have a disease and a diagnosis, but no scientists necessarily working on the project. And how do we kind of bring those resources to bear and work together?
Yeah. So here, I have to admit, I'm not the right person to give very sophisticated answers about therapeutics, because when just to realize that what my community is in the genomics community, we do really well up to the end of where nucleic acids are involved. And of course, where therapeutics are going to pick up, it's going to be very specific to either technologies that relate to therapeutic development, which is not what ours is, or going to be very intertwined with the biology of the disease. And so and in some ways you almost want that. So our success is when we hand off a disease and we say, we now know the genomic basis, and we hand it off to another expert. And that expert uses their biological knowledge of that biochemical pathway or of something that they've learned about, where they finally know what is causing the disease. And I'm not just trying to skirt by it. I'm actually making the point that that's actually a success for us, because what is more valuable for a rare disease than to have graduated from being, well, we have absolutely no idea what the cause is, to at least we know the cause, and now we could bring a whole new set of experts in who understand something about that biological process. And it's not surprising that the person like me who may have figured out what that mutation is the wrong set of expertise compared to the person that's going to really know, oh, I see, you're affecting this biological pathway. I've been studying that for the last 40 years of my life. I can tell you all about it. So, what I'm trying to do is to graduate as many genetic diseases out of my sphere and get them in the hands of people that really understand the nuances of the biological processes involved. And, again, the glass half full, we've gone from 61 to nearly 6,000 rare diseases. We now know the genetic basis. The glass half empty is we still have another 3,000. Why does the Gregor Consortium, why don't we just launch it? To try to fill in those last 3,000.
Mhm, mhm. Thank you for that. So we have just a couple more really great questions. I'd love to ask you if you're willing. We have a couple of questions actually that have touched on internationalization of genomics, and folks asking about how NHGRI is kind of looking forward to bringing in the rest of the world and expanding and making accessible genomics to the rest of the world. So we'd love your thoughts there.
So you're interested in, well, I mean, there's 2 points to be made, and I'm not sure how the question was being asked. Actually, let's take a very rare disease centric view of internationalization. That's just statistics. The fact of the matter is, the more, when you're studying rare, especially ultra rare diseases, the power come in numbers, and the more cases you can get, the more you'll learn about the disease. You'll learn from each other, and it also just helps more solidify evidence to help us advance our knowledge. And if it's ultra rare, you need the whole world's populations, because these things happen a little bit at random. And so the more we can do to interconnect people and patients and physicians around the world to say, oh my gosh, each of us has one patient, and alone we'd never figure out what was wrong with them. When we put the 3 patients together, now we figure out that is very powerful. So I think that is one set of reasons why internationalization is so important in rare diseases. More from a more sort of health equity, social justice point of view, and I've been quite involved in this. We don't want genomics to just be for the rich countries. We don't want genomics just to be for those that have access to the best healthcare. We want to see genomics available to everyone. And so I've been quite involved in some major projects, including in Africa, to capacity build genomics expertise around the world. And of course, ways to really make that, there's a lot of ways to make that happen. First of all, the cheaper you can make things, the more you can make it happen. And the more you can make things accessible on the internet for free, the more you can make that happen. And so I continue to be involved in a number of international organizations to try to do exactly that. Because at the end of the day, we can really improve human health in a lot of ways through genomics, and that would be benefited if we have expertise around the world, not just concentrated in just a few countries.
Yes, thank you for that. And one last question.
Sure.
Someone asked about the issue of data security, and how do we balance all of these exciting advances in genomics with perhaps outdated genetic discrimination laws, and how do we stay ahead of? They ask about a number of different things, but I'm curious about just in general your thoughts about what's the future? What does the future need to look like in terms of data security and privacy in order to be able to realize the vision that you've laid out?
Well, we need to be relentless on all fronts. I mean, we need to make sure our laws are as strong as they can be. So we never should. You got to keep pushing. And any evidence of genetic discrimination should be fought back and prosecuted, and we should, it has to be exposed, et cetera, et cetera. We need to develop better and better computational methods to get us as much privacy as we can get. But I also think we need to recognize that this is not unique, that genomics and genetics is not unique. There's a lot of information about us out there, health information, our financial information. There's a lot on your visa statement that we could figure out about you, and so all of these things need to be protected. And yes, there are some nuances associated with genetics that are a little more complicated, but there is a lot of health information that's not genetics that's pretty complicated. I just think then we need to balance all of that against how much benefit can be achieved. And I would argue that if you talk to people who are benefiting from genomic medicine implementation, they're on balance, they're quite happy about that, and they feel the risks are comparative risks of privacy breaches are pretty small. But it's not to say we can get cavalier about it, and I think that's the last thing we want to be, is cavalier. We need to be cognizant and really vigilant to on all fronts, like I described.
Wonderful. Well, let me just thank you again so much for taking the time to speak with us today, for answering our questions, for that wonderful presentation. And for everyone who's still on, you can learn more about the Undiagnosed Diseases Network and the UDN PEER group on our website. And this will also be recorded. So if you would like to view it again, it will be available on our website within the next couple of days. And with that, we will wrap up, and thank you again, Dr. Green, for your time.
And congratulations to you for organizing this, and good luck with future such lectures in the future. There's a lot of great people you could bring to this.
Wonderful. Thank you so much.
All right. Take care.
Bye-bye.